Exploring Life, Chapter 5. Chemistry of Carbon, Part 2. Biochemistry is a branch of organic chemistry focused on the chemical processes that occur within living organisms. At the heart of this field are four major classes of organic molecules, each essential to life. Carbohydrates, the body's main source of energy. Proteins, responsible for structure, function, and regulation of tissues and organs. Lipids, used for long-term energy storage, insulation, and building cell membranes. Nucleic acids, like DNA and RNA, which store and transmit genetic information. Together, these molecules form the foundation of all biological activity. This table summarizes the four major categories of organic macromolecules, their building blocks, and their polymer forms. Carbohydrates, proteins, and nucleic acids are made from specific monomers, monosaccharides, amino acids, and nucleotides, respectively, and each forms true polymers through repeated bonding of these units. Lipids are a bit different. They're made from fatty acids and glycerol, but they don't form true polymers. Now, let's take a closer look at each of these macromolecules in more detail. Let's start with carbohydrates, and specifically, glucose, a key monosaccharide with the molecular formula C6H2O6. Glucose is the body's primary energy source and plays a central role in cellular respiration, the process by which cells extract energy. Plants produce glucose through photosynthesis, using sunlight to convert carbon dioxide and water into this vital sugar. We get glucose from natural sources like fruits, vegetables, and honey, foods that provide quick, accessible energy. This image shows two forms of glucose, a fundamental sugar molecule. Glucose can exist as both a straight chain and a ring structure. However, in aqueous, or water-based, solutions, glucose predominantly exists in its ring form. That's because the ring structure is more stable and far more common in biological systems, where glucose plays a central role in energy metabolism. So while the straight-chain form is important for understanding how glucose behaves chemically, it's the cyclic form that's most relevant in living organisms. Carbohydrates are made up of building blocks called monosaccharides, or simple sugars. Common examples include glucose, fructose, and galactose. When two monosaccharides join together, they form a disaccharide. For example, table sugar, or sucrose, is made of glucose and fructose. Long chains of monosaccharides form polysaccharides, which are complex carbohydrates. These include glycogen, starch, cellulose, and chitin. Polysaccharides are long chains of sugars that serve different functions depending on their structure. Glycogen is the main energy reserve in animals, stored primarily in the liver and muscles for quick access. Starch serves a similar purpose in plants, it's their primary energy storage molecule. Cellulose provides structural support in plant cell walls. It's tough, rigid, and indigestible to most animals. Chitin offers strength and protection in the exoskeletons of arthropods, like insects and crustaceans, and also in the cell walls of fungi. Though all made of sugar units, their structure determines whether they function as fuel or as framework. Starch, cellulose, and glycogen are all polymers of glucoses built from repeating glucose units. Chitin is also similar, but it's made from a modified form of glucose. What's fascinating is that these polymers differ not in what they're made of, but in how the glucose units are arranged. For example, glycogen stands out because of its highly branched structure, which allows animals to rapidly access stored energy. These subtle variations in orientation and bonding give each polysaccharide its unique function, whether for energy storage or structural support. You can think of it like a jeweler crafting different chain designs, each made from the same type of gold rings, but woven in distinct patterns. The material is identical, but the final product looks and functions differently depending on how it's put together. Carbohydrates aren't just about energy, they also play a critical role in cellular recognition and communication. Two key examples are glycolipids and glycoproteins. Glycolipids are lipids with attached carbohydrate chains. Glycoproteins are proteins with short carbohydrate chains called oligosaccharides. These molecules are found on the cell surface and are essential for recognizing other cells, sending signals, and maintaining proper cell function. In essence, they act like molecular name tags, helping cells identify and interact with one another accurately. Now let's move on to the next major group of macromolecules, lipids. The key characteristic of lipids is that they are insoluble in water, they don't mix well with it. This property is essential to how lipids function in the body. There are several main types of lipids. Fats, or triglycerides, consist of a 3-carbon molecule called glycerol bonded to 3 fatty acids. Phospholipids have a similar structure, but instead of 3 fatty acids, they contain 2 fatty acids and a phosphate group. Steroids are structurally unique, made up of 4 interconnected carbon rings. Despite their differences, all members of the lipid family play vital roles in energy storage, membrane structure, and chemical signaling within the body. Let's take a closer look at fats and their different forms. Saturated fats have no double bonds between their carbon atoms. This makes their structure straight and tightly packed, so they're typically solid at room temperature, like butter. Unsaturated fats have 1 or more double bonds, causing bends in the fatty acid chains. This makes them liquid at room temperature, as seen in oils. Through a process called hydrogenation, unsaturated fats can be converted into saturated fats to improve shelf life and texture in processed foods. However, this process can also create trans fats, a type of fat where the orientation of the double bonds is different from those found in nature. The trans fats have been linked to negative health effects, particularly an increased risk of heart disease. Phospholipids have a unique structure that makes them perfect for building cell membranes. Each phospholipid has 2 hydrophobic tails, which repel water, and 1 hydrophilic head, which is attracted to water. When placed in an aqueous environment, phospholipids naturally arrange themselves into a bilayer. The hydrophobic tails point inward, away from water, while the hydrophilic heads face outward. This phospholipid bilayer forms the foundation of all cell membranes, creating a flexible, semi-permeable barrier that protects and organizes the cell. Here we see a side-by-side comparison of a fat molecule on the left and a phospholipid on the right. Both are built on a glycerol backbone, but their structures differ. A fat, or triglyceride, is composed of 3 fatty acids bonded to glycerol. A phospholipid, by contrast, has only 2 fatty acids, and the third position is occupied by a phosphate group. This phosphate group is what gives the phospholipid its hydrophilic head, allowing it to interact with water, unlike the fatty acid tails, which remain hydrophobic. The R group attached to the phosphate is a variable chemical group that can differ between types of phospholipids, often influencing membrane function or signaling. The phospholipid bilayer is the major structural component of all cellular membranes. Each phospholipid has a hydrophilic head that faces the aqueous environment either inside or outside the cell, while the hydrophobic tails are tucked away in the interior of the bilayer, shielded from water. Because of this dual nature, phospholipids spontaneously form membrane-like bilayers whenever they're placed in water. Let's bring the plasma membrane to life. Each phospholipid has a water-loving head and two water-fearing tails. In cells, they form a bilayer. Heads face the watery inside and outside, while tails tuck together in the middle. Try modeling this with your fingers, arms, or full body, and invite friends to join in. 2 rows, tails in, heads out, that's your living phospholipid bilayer. Steroids are a unique type of lipid, characterized by their structure of 4 interconnected carbon rings. One important steroid is cholesterol, which plays 2 major roles. It's a structural component of cell membranes and a precursor for producing other steroids, including hormones. Sex hormones like testosterone and estrogen are derived from cholesterol. These hormones are essential for regulating sexual development and reproductive function. You might have heard of anabolic steroids, synthetic derivatives of testosterone that also belong to this group. While they can promote muscle growth, their misuse can lead to serious health risks, including liver damage, hormonal imbalance, and cardiovascular issues. This is a great moment to revisit how small structural differences can lead to very different biological roles. Here we see cholesterol and testosterone, both members of the steroid family. You might recognize testosterone on the right. We introduced it earlier when discussing functional groups, alongside estradiol. On the left is cholesterol, with its signature 4-fused ring structure and a hydroxyl group at one end. It serves as both a structural component of cell membranes and a precursor to other steroids. Testosterone shares the same steroid backbone, but includes additional functional groups, like ketones and hydroxyls, that give it distinct hormonal activity. So yes, small differences can make all the difference. Just like the difference between coffee and decaf, same beans, very different results. Proteins are essential macromolecules made up of long chains of amino acids, also called polypeptides. The monomers of proteins are amino acids, and each amino acid has 3 main parts: an amino group, a carboxyl group, and a unique R group that gives it specific chemical properties. Amino acids are linked together by peptide bonds, which are formed through dehydration synthesis, a process that removes a water molecule as the bond forms. These chains fold into specific shapes, allowing proteins to perform a wide range of functions in the body. This figure shows the general structure of an amino acid, the fundamental building block of proteins. At the center is a carbon atom, known as the alpha carbon, which is bonded to 4 main components: a hydrogen atom, an amino group, a carboxyl group, and a variable side chain, called the R group. It's the R group that makes each amino acid unique. There are 20 standard amino acids used in proteins, though more exist, and each has a different R group that gives it specific chemical properties, such as being acidic, basic, hydrophobic, or hydrophilic. Proteins have a highly organized structure made up of 4 distinct levels, each adding more complexity and function. These are the primary structure. the secondary structure, the tertiary structure, and the quaternary structure. Each level builds on the previous one, ultimately shaping the protein's final form and determining how it works in the cell. This image illustrates the hierarchical levels of structural organization within a protein. We start with the primary structure, the linear sequence of amino acids. Next is the secondary structure, showing localized folding into alpha helices and beta sheets. Then comes the tertiary structure, where the entire chain folds into a complex three-dimensional shape, driven by interactions between our groups. Finally, the quaternary structure appears when multiple polypeptide chains come together to form a functional protein complex. Each level builds on the last, and together, they determine the protein's final shape and its function in the cell. A sweater analogy can help us understand the 4 levels of protein structure. The primary structure is like the yarn itself, a continuous thread representing the sequence of amino acids. The secondary structure is the knitting pattern, local coils and folds, like alpha helices and beta sheets, that begin shaping the fabric. The tertiary structure is when the sweater takes form, with sleeves, collars, and curves, representing the three-dimensional folding of a single polypeptide chain. And the quaternary structure is the complete sweater, formed when multiple polypeptide chains are stitched together into a functional protein. Just like a well-knit sweater, a protein's structure determines its function, and it all starts with the right thread. Imagine taking a beautifully knit sweater and tossing it into a hot wash cycle. It shrinks, twists, and loses both its shape and function. Proteins can go through something very similar in a process called denaturation. This occurs when factors like heat, changes in pH, or shifts in salt concentration disrupt the delicate bonds that maintain the protein's structure. And just like a misshapen sweater can't keep you warm, a denatured protein can no longer perform its role in the cell. Now let's look at the final group of macromolecules, nucleic acids. There are 2 main types, DNA and RNA. Both are made of monomers called nucleotides, and each nucleotide consists of 3 parts, a sugar, a phosphate group, and a nitrogenous base. In DNA, the 4 bases are adenine, thymine, cytosine, and guanine. In RNA, uracil replaces thymine, but the other bases remain the same. These molecules are essential for storing, transmitting, and expressing genetic information, the instructions for life. While ATP isn't classified as a nucleic acid, it is a nucleotide, and it's closely related to this group. Because of its importance, we'll take a moment to describe it here. This illustration shows the molecular structures of DNA nucleotides, RNA nucleotides, and ATP, a molecule that plays a key role in both energy transfer and nucleic acid synthesis. ATP, or adenosine triphosphate, has a dual role. It provides the energy needed to drive the chemical reactions that build DNA and RNA, and it also contributes essential components. Its adenine base and phosphate groups are incorporated into the structure of nucleotides, the building blocks of DNA and RNA. So, ATP isn't just the cell's energy currency, it's also part of the blueprint itself. Let's look at the structural differences between DNA and RNA. DNA has a double helix structure, 2 strands twisted around each other. Each strand has a sugar phosphate backbone, and the strands are held together by base pairs, adenine pairs with thymine, and cytosine pairs with guanine. RNA, on the other hand, is typically single-stranded. It also has a sugar phosphate backbone, but instead of thymine, uracil pairs with adenine. These structural differences also affect stability. DNA's double-stranded structure and deoxyribose sugar make it more chemically stable, ideal for long-term genetic storage. RNA, with its single strand and ribose sugar, is less stable, which suits its temporary roles in information transfer and protein synthesis. This illustration shows the double helix structure of DNA. Each individual strand is stabilized by covalent bonds between adjacent nucleotides, forming a strong sugar phosphate backbone along the length of the strand. The two complementary strands are held together by hydrogen bonds between the nitrogenous bases, adenine pairs with thymine, and guanine pairs with cytosine. These hydrogen bonds add stability to the overall structure and play a crucial role in ensuring the accurate transmission of genetic information during DNA replication and protein synthesis. This illustration we'll explore this in more detail later in the course, but here's a quick overview of how genetic information is transferred. First, the information stored in DNA is copied into RNA through a process called transcription. Then, that RNA is used to build proteins during translation. A gene is simply a section of DNA that carries the instructions for making a specific protein. It determines the sequence of amino acids in that protein. Together, these steps form what's known as the central dogma of molecular biology: DNA, RNA, protein. And if you're wondering why it's called a dogma, well, it's like a law on steroids. In biology, this is the core principle that everything else builds on. ATP, or adenosine triphosphate, is not a nucleic acid but a nucleotide, which is why it is discussed here. It serves as the cell's primary energy carrier, essential for nearly every process that sustains life, from building molecules and transporting substances to driving cell division and communication. Without ATP, cells would lack the energy required to function. ATP, shown on the left, is composed of a nitrogenous base, adenine, blue, ribose, purple, and 3 phosphate groups, yellow. Structurally, it closely resembles an RNA nucleotide, specifically adenosine, shown on the right, except that ATP has 3 phosphate groups, whereas RNA nucleotides have only 1. Not surprisingly, ATP plays a dual role. It supplies the energy needed to power the chemical reactions that build DNA and RNA, and it also contributes essential structural components, namely, the adenine base and ribose sugar, which are identical to those found in RNA nucleotides. Let's look at how ATP transfers energy. It all centers on the 3rd phosphate group. When energy is needed, ATP loses a phosphate through hydrolysis, releasing energy and forming ADP. The freed phosphate can then attach to another molecule, phosphorylating and energizing it. When energy becomes available again, ADP is recharged into ATP through dehydration synthesis. Enzymes catalyze each step. Think of ATP as a prepaid energy card. The cell spends a bond for energy, then recharges it by adding the phosphate back. Isn't it kind of amazing that the chemistry of a single element, carbon, took more time to explore than the formation of the entire universe? It was a lot to take in. We even had to split the lecture into 2 parts. And let's be honest, you probably needed a few breaks along the way. I know, it's super cool and it's also challenging. But you made it this far, and I'm genuinely proud of you. Now it's time for review. Feel free to take notes and move back and forth through the slides as you answer the questions. Don't rush, take your time, and really reflect on everything you've learned. Isn't it kind of amazing that the chemistry of a single element, carbon, took more time to explore than the formation of the entire universe? It was a lot to take in. We even had to split the lecture into 2 parts. And let's be honest, you probably needed a few breaks along the way. I know, it's super cool and it's also challenging. But you made it this far, and I'm genuinely proud of you. Now it's time for review. Feel free to take notes and move back and forth through the slides as you answer the questions. Don't rush, take your time, and really reflect on everything you've learned.